Good morning all. This is the second class in first chapter. Yesterday we discussed about what are the autotrophs, what are the heterotrophs, and how the plants are preparing their own food by the using of photosynthesis process. And also we discussed about photosynthesis equation and completed one experiment. It is testing of starch in the leaves, presence of starch in the leaves. So today also we will discussing different kind of topics in first chapter. Before going to start, I want to ask some question. Write in your notebook and also write answer for these questions. What are the end products of photosynthesis? Second question, which solution is using for testing of starch? Write the equation of photosynthesis. Name the factors of photosynthesis. And last, fifth question, how can you test the starch present in the leaves? Answer the questions. Commonly, all the plants are using water to producing glucose in the plants in the photosynthesis process. But some bacteria, especially purple sulfur bacteria, using H2S, hydrogen sulfide, instead of water. So it is producing sulfur gas. Commonly, all the plants are using H2O, water, for production of glucose in the photosynthesis, producing oxygen. But some bacteria, example, purple sulfur bacteria, using H2S and producing sulfur gas instead of oxygen. And also, one helmet find the water essential for the increasing of plant mass. Water also important for increasing of plant mass. Joseph Priestley in 1770 performed a series of experiments that revealed the essential role of air on the growth of green plants. Joseph Priestley conducted a series of experiments in 1770. So he found the main important role of the air in green plants. What is the air? What is the importance of air in the plant growth? It is discovered by the Joseph Priestley. After that, in 1774, Joseph Priestley discovered oxygen gas, and also it is named by Lavoisier in 1775. Priestley observed that candle burning in a closed bell jar. When we are seeing this picture, one candle is closed with bell jar. When we are closing candle with a bell jar, it is exhausted. In the second observation, we will see we are closed one mouse and candle with using of bell jar. In this, we will observe candle put off and mouse is suffocated. And after Priestley introducing one mint herb into the bell jar, he found that the mouse stayed alive and the candle, when lighted from the outside, and continued burning in the presence of the mint plant. Finally, Priestley concluded follows: plant restore the air what burning animals and burning candles remove. So whenever we are using candle and mouse, they are using oxygen present in the bell jar, and it is decreasing oxygen percentage. Candle will be put off and mouse also suffocated. So when we are using mint plant in the bell jar, it is producing oxygen. So this oxygen used by the mouse and also candle, so both are in normal state. So in this activity, Priestley concluded that so plants are producing oxygen this oxygen used by the candle and mouse. Candle and mouse producing carbon dioxide, it is used by the plant. So interdependence we can see in this here activity. So oxygen, carbon dioxide. Carbon dioxide is important for the plant food synthesis, and it is producing oxygen. So here we can confirm that carbon dioxide is essential for photosynthesis. Oxygen is important for respiration. Massive amount of gases exchange occurs through the stomata as long as they are open, while plants also carry on gases exchange through the loose tissues in the stems and roots, etc. So massive amount of gases exchange occurs through the stomata. Stomata is a plant nose. With using of stomata, plant exchange the gases. So we already know there are 4 factors are important for the photosynthesis. One is carbon dioxide, one is water, chlorophyll and light. If any factor is absent for the photosynthesis, there is no preparation of glucose in the leaves. So we will test the essential of carbon dioxide. Carbon dioxide is necessary for photosynthesis. We will test with one small activity. With using of small activity, we will prove carbon dioxide is important for photosynthesis. For this activity, you need some different kind of operators. First, potted plant, wide mouth bottle, KOH solution, spilt cork, iodine solution, petri dish, and rubber cork. These are the operators you have to use in this activity. Procedure. First of all, you have to take one wide mouth bottle, put potassium hydroxide pellet or potassium hydroxide solution in the bottle. Potassium hydroxide solution is useful for absorption of carbon dioxide. It is present in the bottle. So what is the use of KOH? KOH is useful for absorbing carbon dioxide. It is present in the wide mouth bottle. After that, you have to take one potted plant. Before using this plant, you have to keep this plant in the dark room for 1 week. By placing the plant in a dark room, the starch is removed from the leaves. When we are using fresh plant, some amount of starch present in the leaves, it is interfere our result. So you have to keep plant in the dark room for 1 week. So it is removing starch present in the leaves. After that, you have to take out the plant with using of spilt cork. With using of spilt cork, you have to insert the off leaf inside of the bottle and off leaf outside of the bottle. And after this total plants you have to keep into the sunlight. After a few hours, test the leaf and any other leaf of this plant. You have to test the 2 leaves. One is leaf with bottle and second one is normal leaf. The leaf part which is exposed to the atmosphere, air and light, it is becomes blue black color, and the another side it is present in the bottle, it is does not turns into the blue black color, it is remain colorless. This proves that carbon dioxide is necessary for photosynthesis. In this activity, we have to conclude carbon dioxide is necessary for photosynthesis. Once again, I want to repeat this activity. First of all, you have to take one wide mouth bottle and with using of spilt cork off leaf, you can arrange it into the bottle and off leaf outside of the bottle and fix the cork very tightly into the bottle. And after, you have to keep these total operators into the sunlight. After few hours, you have to take out the leaf and test for the starch with using of iodine solution. When we are testing starch in the leaf with using of iodine solution, off leaf turns into the blue black color. It is indicating starch, especially outside leaf turns into the blue black color. And off leaf present in the bottle, it does not change into the blue black color because of in a bottle we are using KOH solution, so it is not receiving carbon dioxide inside off leaf present inside of the bottle. It does not receiving carbon dioxide, so it is not performing photosynthesis. That's why it's not turns into the blue black color. Outside part of the leaf, it is turns into the blue black color. So we will confirm carbon dioxide essential for photosynthesis. In this activity, you have to take some precautions. First one, when we are inserting leaf into the bottle, that inside bottle leaf does not touch to the KOH solution. First precaution, when we are inserting leaf into the bottle, that inside bottle leaf does not touch to the KOH solution. And second precaution, when you are fixing cork into the bottle, you have to fix airtight. Outside air does not enter into the bottle. So these two precautions you have to take care. So this is our activity. In this activity, we will conclude carbon dioxide essential for photosynthesis.